Basic medical sciences, welcome. In this video, I'm going to present on red blood cells, also known as erythrocytes. Right, so erythrocytes or RBCs are large in number compared to the other blood cells like white blood cells and platelets. The number of RBCs ranges between 4 and 5.5 million cubic millimeters of blood. Right, but there are some units which are easy, like in your manuals. If you're in Ukraine, you use the number per liter of blood. Right, for example, in adult males, it's 4.5 to 5.5 times 10 to the power of 12 per liter, and in adult females is 4 to 4.5 times 10 to the power of 12 per liter. Right, so these numbers, it varies like with the source you are using. For example, if you find a woman with 3.8 or 3.9 times 10 to the power of 12 per liter, that can be normal also, right? So you just need to know the kind of source you are using at your university and all will be fine. Normally, the red blood cells are disc-shaped and biconcave, right? Or a dumbbell. Can you see this? It looks like a dumbbell, right? So this biconcave shape has a very important significance, right? So what is the significance of biconcave shape of the red blood cells? Firstly, the biconcave shape helps in equal and rapid diffusion of oxygen and other substances into the interior of the cell. This shape provides large surface area, right? So large surface area is provided for absorption or removal of different substances from and out of the red blood cell. Minimal tension is offered by this shape on the membrane when the volume of the cell alters, right? Because of biconcave shape, while passing through minute or small capillaries, the RBCs squeeze through the capillaries very easily without getting damaged, right? Okay. Red blood cells, they don't have nucleus, right? They are non-nucleated. The only mammal with nucleated RBCs is camel, right? So camel have nucleated red blood cells. Because of absence of nucleus in human RBC, it means DNA is also absent under normal conditions. The organelles such as mitochondria and Golgi apparatus are also absent in the red blood cell. So how does this cell manufacture energy, right? So the energy is produced from glycolytic process or glycolysis, anaerobic, not aerobic, but anaerobic glycolysis. The red blood cell does not have insulin receptor, so glucose uptake by this cell is not controlled by insulin. Right, continuing with the characteristics. The RBCs has a special type of cytoskeleton which is made up of actin and spectrin. Both of these proteins, actin and spectrin, are anchored to membrane proteins by means of another protein called ankyrin. Right, so this is the cell membrane of the RBC. Right, so right, this is the lipid bilayer, right? These are the two proteins, right? You can see like the alpha chain and beta chain. So this is spectrin, alpha and beta spectrin, and this is ankyrin, right? So it anchors like this spectrin to other proteins on the cell membrane. The absence of spectrin results in hereditary spherocytosis. So in this condition, hereditary spherocytosis, the cell is deformed and it loses its biconcave shape and it becomes globular or spherocytic, like a sphere, right? So the spherocyte is very fragile and is easily ruptured or hemolyzed in hypotonic solutions. Right. The properties of red blood cells. Number one is rollers formation, right? So when blood is taken out of the blood vessels, the RBCs, they have this ability to pile up one above the other, right? Like a pile of coins, right? So this property is accelerated by plasma proteins such as globulin and fibrinogen, right? So it looks like this, right? So you can see like piles of coins, right? That's rollers formation, rollers formation. Okay, that is the first property. The specific gravity of RBCs is 1.092 to 1.101. Packed cell volume, right? So packed cell volume is the proportion of blood occupied by RBCs expressed in percentage, right? So it is also known as hematocrit value. Right, so this value is 45%, and the remaining 55% is plasma volume. Suspension stability. During circulation, the red blood cells remain suspended uniformly in the blood, right? So it's another important characteristic or property, suspension stability, right? So the fate of RBCs. The average lifespan of a red blood cell is 120 days. After that, red blood cells are destroyed in the reticuloendothelial system, specifically the macrophages in the spleen. Okay, so let's see what happens to the aged red blood cells. Right, after their destruction in the spleen, you can see the release of hemoglobin. Right, hemoglobin have three important things: iron, globin, and porphyrin rings. Right, let's start with iron. Iron is combined with a protein known as apoferritin, and they form ferritin. They can either be stored and reused, if you know, like as hemosiderin, like as a storage form. Right, that's the iron pathway. For the globulin will be, they are proteins, right? So they can be broken down into amino acids, as protein pool. They can either be stored or reused again. Then porphyrin rings. Porphyrin can run down different pathways until reaching bilirubin, and then bilirubin is usually excreted, right? So let's follow this pathway a little further. Right, I hope you can see this, right? So again, hemoglobin, in this case, we have ferritin and hemosiderin, the one I mentioned here, right? And on the other side, we have biliverdin. Biliverdin converted to bilirubin. Bilirubin is combined with globulin. Globulin is a transport protein, right? So it goes to the liver as unconjugated, and in the liver there will be conjugation, right, making it water soluble, right? From there are different enzymes important for conjugation, right? We'll talk about them later in another video, right? So this conjugated bilirubin can go into the GIT, and from there it can be released as stercobilin, right? Because stercobilin comes from conversion of stercobilinogen to stercobilin by the bacteria in your gut, right? So there's another pathway like where this bilirubin is cycled back to the liver. It's called enterohepatic circulation, right? So that's the first pathway into the what? Into the stools. Then the other pathway is this bilinogen will go to the kidney, right, and will be converted to urobilinogen and then urobilin, giving the urine a specific color. We'll talk about that in another video, right? Okay, so let's continue with RBCs, right? What are the functions of red blood cells? Number one is transport of oxygen from lungs to the tissues. Hemoglobin in the RBCs combines with oxygen to form oxyhemoglobin. About 97% of oxygen is transported in blood in the form of oxyhemoglobin. Number two, transport of carbon dioxide from tissues to the lungs. Hemoglobin combines with carbon dioxide to form carboxyhemoglobin, or some books say they just say carb hemoglobin, right? And about 30% of carbon dioxide is transported in this form, right? All right, so this is another pathway of transport of carbon dioxide. It is through like as bicarbonate. So carbon dioxide combines with water in the presence of the very important enzyme called carbonic anhydrase to form carbonic acid, which will dissociate to give bicarbonate and protons, right? So it will be transported as bicarbonate, and then the reaction is reversible. That's how we have our carbon dioxide out. It's another way, right? So about 63% of carbon dioxide is transported in this form. Third function, the buffering action, right? So it is an important, the RBCs have an important buffering action in the blood, right? So by this action, it regulates the hydrogen ion concentration and thereby plays a role in the maintenance of acid-base balance. Okay, this is a role in determination of blood group. Okay, let me find a place here. Right. So on the cell membrane of RBCs, we have antigens like antigen A, antigen B, and antigen D. D is rhesus, right? So these are the typing blood groups. They are determined by the kind of antigen which is present on the cell membrane of the RBC, right? For example, if the patient have antigen A, it means this patient is a type 2 blood group, and in the plasma this patient will have what like a beta, yeah, it's called beta antibodies in the serum, beta, okay, or anti-B. We will talk about that later. I just gave that one as an example, but just to know, the blood groups are determined by the kind of antigen which is present on the cell membrane of the red blood cell. Wow, this is about variations in the number of red blood cells, right? So they can be an increase or a decrease. Okay, let me give everything here. Okay, cool. There can be an increase in RBCs and decrease in RBCs, right? And under such circumstances, they can be physiological reasons or pathological reasons, right? Or physiological variations and pathological variations, right? Okay, let's start with a physiological increase in RBC count. The increase in the number during this condition is marginal and temporal, and it occurs in the following conditions. Number one, age. At birth, the RBC count is 8 to 10 million cubic millimeters of blood. And then the count decreases within 10 days after birth due to destruction of RBCs, causing physiological jaundice in the new babies. However, in infants and growing children, the cell count is more than the value in adults. Number two is sex. Before puberty and after menopause, in females, the RBC count is similar to that of males, right? So before, let's just say, okay, I'm going to use puberty from 13, right? So from 13 until the onset of menopause, it will be different, but before 13 and after menopause, it will be same. right? So during reproductive period in females, the count is less than that of males, right? So it's usually less than 4.5 million per cubic millimeters. Number three is high altitude, right? In mountain climbers, right? What happens is that there will be hypoxia. Hypoxia will cause a stimulation of kidneys to produce a hormone called erythropoietin. Erythropoietin will stimulate the red bone marrow to increase the production of red blood cells. We are still talking about physiological variations. We're talking about emotional conditions and muscular exercises. Both these conditions will increase the sympathetic activity. This will increase the secretion of adrenaline and causing contraction of the spleen, right? So spleen, it normally stores a lot of RBCs, right? So because of emotional conditions or like anxiety, for example, or muscular exercise, the spleen will release the RBCs, causing physiological increase in the what? In the RBCs. Number six, increase in environmental temperature. Right. So increase in environmental temperature will increase the RBC. How? In general, the increase in temperature will increase a lot of activities in the body, in all metabolic activities, therefore increasing the production of RBCs. Then after meals, right? So there is a slight increase in the red blood cells after taking meals, and this is because of the need for more oxygen for metabolic activities. Now let's talk about physiological conditions where the red blood cells are decreased. Number one, high barometric pressures. At high barometric pressures, as in deep sea, when oxygen tension in the blood is higher, the RBC count will decrease. Number two, during sleep. The RBC count decreases slightly during sleep and immediately after getting up from the sleep. And generally, all activities in the bodies are decreased. During sleep, including production of RBCs. So this point is more like the opposite of high temperature. Right? In high temperatures, the metabolic processes will increase, and during sleep all the metabolic processes will decrease, including the production of RBCs. During pregnancy, the RBCs also decrease, and it is because of increase in extracellular fluid volume. So increase in extracellular fluid volume increases the plasma volume, also resulting in hemodilution. So there is a relative reduction of RBC count. Now let's talk about pathological variations, right? So you have polycythemia as the increase in RBC count, and anemia is the decrease in RBC count, right? So in this video, we are only going to talk about polycythemia. Anemia, we have a separate video, right? Pathological polycythemia is the abnormal increase in the RBC count, and in most cases is above 7 million per cubic millimeters of blood. And there are two types of polycythemia. We have primary polycythemia and secondary polycythemia. Right. Let's start with primary. Primary polycythemia, also known as polycythemia vera or Varus disease. Right, so this is a disease characterized by persistent increase in RBC count above 14 million per cubic millimeters of blood, and is almost always associated with increase in the white blood cell count above 24,000 per cubic millimeter of blood. Polycythemia vera occurs in myeloproliferative disorders like malignancy of the bone marrow. All right. Okay. So this is important. Primary and secondary polycythemia. I will talk about secondary later, but I just need to mention it now. Primary in the bone marrow, secondary in the kidney. That's all. That's how you start the whole differentiation thing, right? So let's look at the secondary polycythemia, right? This secondary polycythemia, for starters, is started by conditions which causes hypoxia, right? So if you remember, we said hypoxia will stimulate the kidney to produce erythropoietin, and then erythropoietin will go to the bone marrow and cause a production of RBCs, right? And some of the conditions which causes hypoxia, therefore leading to increased stimulation of the bone marrow by the erythropoietin, include the following: respiratory disorders like emphysema, congenital heart diseases is a condition associated with hypertrophy of the right ventricle and obstruction of blood flow to the lungs. Chronic carbon monoxide poisoning, poisoning by chemicals like phosphorus or arsenic, and it also occurs in repeated mild hemorrhages, right? Okay, so there is another condition which I want to mention. It's not a medical condition usually, right? So, athletes, right? They, a long time ago, they used to, it's called, the process is called blood doping. They take out their blood before sports. They store the blood, and then like hours before going to marathon or before taking part, they will re-inject the blood, right? So they will have increased erythrocytes as well, right? So because of this increase in erythrocytes, the blood viscosity will be increased, right? The blood will be thick, causing high blood pressure, right? It's another mechanism. We'll talk about it later, right? That was a variation in count. Now, variation in the size of RBCs. Under physiological condition, the size of RBCs in venous blood is slightly larger than those in arterial blood, right? And in pathological conditions, there are variations in the size of RBCs. They can be micro size, micro for smaller cells, macro size, larger cells, and number 3, anisocytes, the cells with different sizes, right? So again, we have microcytes, macrocytes, and anisocytes are usually present in iron deficiency anemia, prolonged forced breathing, and increased osmotic pressure in the blood. Macrocytes will be present in megaloblastic anemia, decreased osmotic pressure in the blood, and anisocytes, right? The anisocytes will occur in pernicious anemia, right? So I just want you to remember during studying, you need to differentiate. Okay, so the condition will be called anisocytosis, right? You need to differentiate physiological and pathological anisocytosis, right? There are some physiological conditions where you have these anisocytes, right? You need to differentiate it. If I remember, it's about 18%, right? So if there are like more than 18% of cells with different sizes, that will be pathological. Less than is physiological, right? And so on. But please make sure you check that concept because it might come in the MCQs about the size, right? The shape of RBCs is altered in many conditions, including different types of anemias, right? So in terms of variations in shape, so these are the conditions. There is crenation. Crenation is shrinkage, as in hypertonic conditions. Spherocytosis, I mentioned it, is a globular form, as in hypotonic conditions. Elliptocytosis, that's elliptical shape, as in certain types of anemia. Sickle shape, crescentic shape, as in the sickle cell anemia. and poikilocytosis. Poikilocytosis is an unusual shape due to deformed cell membrane, and the shape will be of a flask, hammer, or other unusual shapes, right? And again, you need to be able to differentiate physiological and pathological poikilocytosis. Right, now variations in the structure of RBCs. There is punctate basophilism or punctate basophilia. This is striated appearance of RBCs by the presence of dots or basophilic materials. They are porphyrins, right? They look like this, right? So inside the RBCs, you will find those blue dots, right? This condition mainly occur in lead poisoning. Rings in red blood cells, right? So the rings or twisted strands of basophilic material appear in the periphery of RBCs, right? This is also known as goblet ring. There is another name, they call it Corbett ring. Yeah, I'm sure it's Corbett ring. And this appears in certain types of anemia, and is this eight-shaped is like an eight-shaped string, right? The next one is Howell Jolly bodies. Howell Jolly bodies, right? Forgive my pronunciation, right? But this is another condition in some types of anemia where you find nuclear fragments inside the ectoplasm of the RBCs, right? So, like here. For now, don't worry about this, because in the next video, I'm going to cover into great details the conditions associated with different morphologies, right? So the video title will be Abnormal Red Blood Cell Morphologies and Associated Diseases. Until next time, guys.